Welcome. I'm Stephen Faraone from SUNY Upstate Medical University, here to talk to you about what I've learned in 3 decades investigating the genetics of ADHD. We'll start with the simple fact that we now know that much of ADHD's etiology is coded in the human genome. How do we know that? Well, first let's start with twin studies. There are now 38 studies of twins, these are identical and fraternal twins, that have looked at either the diagnosis of ADHD or symptom counts in the population, and we have one family study of ADHD. And when they compute a heritability, they come up with a mean figure across 38 studies of 74%. Now that's about as high as schizophrenia or bipolar disorder, which makes ADHD about as heritable as those other psychiatric conditions. What's very interesting about these twin studies is that they've been done in Europe, Scandinavia, the United States, and Australia. And so that argues against the idea that ADHD is somehow an American condition. In fact, wherever we look for ADHD, we find it, and wherever we assess its heritability, it's fairly high. So what does that mean? Well, the heritability of 74% means we can explain 74% of ADHD's etiology by genes and their interaction with environmental risk factors. The second lesson I've learned is that ADHD, and in fact most common forms of psychopathology, are polygenic. And by polygenic, I mean many DNA variants. These are variants in the genome that increase the risk for ADHD. And I use the term variants because we don't know that all of these variants are in genes. In fact, some of them are in regulatory regions that control the expression of genes. These are the results from our very big study, almost 50,000 people, studying the genetics of ADHD in a genome-wide association study. In this type of study, we look at literally millions of genetic markers across the genome to find spots that are significantly associated with ADHD after correcting for the fact that we did so many statistical tests. Now, in this plot, which is called a Manhattan plot, the higher values are more significant, and the dotted line is the threshold of genome-wide significance. And the important finding here in this paper in Nature Genetics, which is now published, is that 12 loci were genome-wide significant. So this is the first firm evidence we have of genomic loci involved in ADHD. We also found evidence for a highly significant polygenic risk score. What does that mean? What this means is that in our statistical analyses, although we could only confirm 12 genome-wide significant loci, we could confirm that there must be hundreds, if not thousands, of other loci that also increase the risk for ADHD, but we could not detect their individual effects, we could only detect their joint effect. And because of that, we can create a polygenic risk score, which indexes the number of ADHD risk alleles carried by an individual. So everybody in the population has some ADHD risk variants. People who have too many of them, they would be the people in red on the slide, will have ADHD. People who have fewer may have a few ADHD symptoms. They may be mildly inattentive, mildly hyperactive. People at the low end, near 3 or 3.5, have no ADHD symptoms. Now, this particular distribution is just a cartoon to help you understand the distribution of risk in the population. So why is this polygenic risk score important? Well, given polygenic risk scores, we can now correlate the polygenic risk between 2 disorders or between a disorder and some other condition or some other psychological trait. So I'll be talking a bit about genetic correlations in this talk, and when I use the term genetic correlation, I mean the degree to which 2 disorders share common DNA variants. In this lecture, I won't be talking about rare DNA variants, variants that may occur 1 in 1,000. There are a few that are known to cause ADHD, but the study of rare variants in ADHD is still in its infancy, so we won't be talking about it today. One of the remarkable findings from our Nature Genetics paper occurred when we looked at the correlation between the DNA variants that cause ADHD and DNA variants that are responsible for ADHD symptoms in the population. And this was based on comparison with a study known as Eagle-Kimmer, which studied 20,000 children and adolescents and simply had their parents rate them in terms of the number of ADHD symptoms. When we correlated the DNA variants between our ADHD GWAS and the Eagle-Kimmer GWAS, the correlation was 0.97. It was not significantly different from 0. And that tells us that the genetic risk for the diagnosis of ADHD is essentially identical to the genetic risk that causes the distribution of symptoms in the population. It's an extremely important finding because it confirms data from other twin studies indicating that ADHD is a dimensional trait in the population rather than a yes or no diagnosis. Now, the third lesson that I've come to understand very well from recent studies, recent molecular genetic studies, is that ADHD shares much of its genetic etiology with other psychiatric disorders. Now, this slide is a little bit complex, but let me help you understand it. At the very top, you see different disorders listed: schizophrenia, depression, bipolar disorder, ADHD, autism spectrum disorders, Tourette syndrome, anorexia nervosa, and obsessive compulsive disorder. Now, I've boxed off the correlations for ADHD. If there's an asterisk in the box, it means that the correlation between the two disorders is statistically significant. The darkness of the color tells you how large the correlation is. So, for example, a very dark blue would mean a very large positive correlation, and a dark orange would be a large negative correlation. And as you can see, ADHD has a significant correlation with almost all of these disorders. And most of these correlations are positive. They're small, but they're positive and statistically significant. This set of data is very interesting to me because back in the 1990s, myself and others did studies of clinical referrals with ADHD, and we found extensive comorbidity with other disorders. This was actually a big controversy for a while. And after many years of clinical studies and epidemiologic studies, we realized that this comorbidity was real. And now, for the first time, we know that this comorbidity also has a source in a common genetic etiology. Now, we're not saying that all the DNA variants that ADHD has are shared with these other disorders, because, in fact, the genetic correlations tend to be low. What we're saying is that some of the genetic etiology is shared, and that accounts for the comorbidity that is seen in clinical samples and in populations. The fourth lesson I've learned in my studies of ADHD and reading the literature is that ADHD shares polygenes with psychological traits and even medical conditions. Here again, I'm showing you more genetic correlations, and this time they're on the horizontal axis. Each of those dots in the graph tells you the magnitude of the genetic correlation, and then the horizontal bar is the 95% confidence interval. If you look at the red dots at the top of the graph, you can see that ADHD is negatively correlated, or I should say the genetics of ADHD is negatively correlated with the genetic background for childhood IQ, years of schooling, completing college, intelligence testing, and measures of verbal and numerical reasoning, and also having completed a college degree. But what does that mean? What it means is that, for example, somebody did a study of the genetics of IQ. And what they showed was that there was a polygenic background, polygenic risk for IQ, DNA variants that increased the risk for IQ. Increased the likelihood that you have a high IQ. And what we did is we correlated those DNA variants with ours, and we found out that the DNA variants that are correlated with having a high IQ are actually more likely to lower your risk for ADHD. So there's a negative genetic correlation. That's consistent with the fact from the clinical literature that people with ADHD tend to have a lower IQ score, on average 9 points lower than people without ADHD. And then you can see at the bottom of the graph other correlations with depression, anorexia nervosa we've already talked about. Quite interesting is the negative correlation with subjective well-being. This means that the variants that lead to subjective well-being are also variants that lower your risk for ADHD. And that's consistent with the idea that ADHD people have a lower quality of life, tend to have a lower level of self-rated happiness than other people. When we looked at other traits, we see ADHD being genetically correlated with smoking cigarettes, interestingly also correlated with lung cancer, which makes sense given the cigarette smoking. Some curious correlations in the middle there in brown. The genetic variants that increase risk for ADHD are negatively correlated with genetic variants that determine how old you are at the birth of your first child, meaning that if you have a genetic risk for ADHD, you're also genetically at risk, if you will, to have children at a younger age. You're also more likely to have not only children, you're also more likely to have more children born to you if you have ADHD, because the genetic risk between the number of children and ADHD is positively correlated. Both of these findings are consistent with what we know in the literature about when ADHD people tend to have their first children and the number of people in families of ADHD parents. The findings about age of death are very consistent with recent data in the literature showing that ADHD increases the risk for premature death. In the data you see here in green, we have compared our genetic data with genetics from a study that looked at the genetics of parental death. That's a study which simply asked how old was your mother when she died, how old was your father when he died. And that is a somewhat genetically predictable trait. It turns out that the genetic variants that predict early or premature mortality are the same genetic variants that are associated with ADHD, again, consistent with the clinical data. Here's just one example of an association of polygenic risk with ADHD. This is the association of ADHD and body mass index. There's a well-known association between ADHD and obesity in the clinics and in the population. And here we see that as the polygenic risk for ADHD increases, that's what we see on the horizontal axis, the risk for high BMI also increases fairly dramatically. And as you can see, the statistical significance level is exceedingly high, meaning we can be very confident that this is a positive result. And the same is the case with the other genetic correlations I've shown you. These are all highly statistically significant. So what do we know about the DNA variants that we found in our studies of ADHD? Well, it's very clear that these variants implicate genes that are expressed in the brain, and that many of them are also in areas that are involved in regulatory elements that control the expression of other genes. You can see in this graphic here where we're listing different tissue groups on the horizontal axis, and then we're listing the probability that genes from that tissue group are enriched in our ADHD genetic findings. Anything above that green line is statistically significant. As you can see, it's only genes, or I should say DNA variants, that are expressed in the central nervous system that were significantly enriched in the ADHD data. And that's very consistent with the current hypothesis that ADHD is indeed a brain disorder, very consistent with findings from neuroimaging studies showing neuroimaging abnormalities in both children and adults with ADHD. We can also subdivide the data in a different way, looking at what we call functional annotations. What are the functional features of some of these loci? And here, the significant findings are all in, I shouldn't say all, mostly in regions that are involved in epigenetics or the regulation of the genome by the environment. We also find that the highest bar is for DNA variants that are in what are called conserved regions. And those are regions of the genome that haven't changed very much during evolution, and that means they're very important for survival. They don't change, they don't have mutations, because if they do, they typically will kill the individual. They won't survive to reproduce. So we do know now that ADHD variants tend to be highly expressed in the brain, in highly conserved regions, and many of them are in regulatory regions involved in epigenetics and the regulation of gene expression. A sixth lesson from ADHD genetics is that the polygenes that we're discovering for childhood ADHD are very similar, not identical, to the polygenes we're discovering in studies of adult ADHD. Now, these data here, which are currently submitted for publication, looked at two different data sets. The first was from the Psychiatric Genomics Consortium, or PGC. It was a subset of those data that only came from sites that were recruiting children with ADHD. And then the other was from the Impact Consortium, and those data came from sites that were recruiting adults with ADHD. Now, as you can see from the slide, both childhood and adult ADHD have a SNP heritability of about 0.2. So what is SNP heritability? Well, you remember I told you that the heritability of ADHD was 74%. Well, these SNP heritabilities of about 20% tell us that the heritability that's due to common genetic variation is 20%. Now, that's less than the heritability from twin studies, which means there's a gap that we need to explain. And what explains that gap? Well, it's probably due in part to rare variants, but it's also possibly due, and likely due, I should say, to gene-gene interaction and gene-environment interaction. Another finding from this study is that the genetic correlation between ADHD when ascertained in childhood and ADHD when ascertained in adulthood is 0.8. Now that's not perfect, it's not 1.0, and that tells us that there are some DNA variants that likely account for the persistence of ADHD into adulthood, and will tell us why some people remit with the disorder. We know about 2/3 of ADHD children will go on to have ADHD in young adulthood, but about 1/3 will lose not only the symptoms of ADHD, but also the associated impairments. This is an area for future study to figure out which genes control the persistence of the disorder. In this slide, I'm showing you on the horizontal axis we've recorded the genetic correlation between ADHD and a number of different traits, some of which I've already shown you in other slides. And we also have the genetic correlation for adults in the vertical axis. It turns out that those two genetic correlations are themselves highly correlated, almost perfectly correlated. And that tells us that, if you will, the genetic architecture of ADHD in children and adults is very similar. So what do these data tell us about diagnostic and clinical practice? What do they tell us about what clinicians should do in the clinic? We're still at the very early stages, but we can make some inferences that I think are useful in clinical practice. First, a common question I get is, can we use DNA to make psychiatric diagnoses? The answer here is clear. No, not for the common forms of ADHD. There are some rare variants where if you have a rare mutation, we're pretty sure you'll develop ADHD, but those are very rare and not very helpful in the average clinical practice. The current polygenic risk scores that we can compute are very useful in research, and we've shown some very fascinating associations in the genetic literature, but they're not ready for use in the clinic. We do expect that the accuracy of these scores will improve as our samples get larger, because that reduces the noise in the score. They'll improve as we use more sophisticated algorithms, such as artificial intelligence, machine learning. And they'll also improve as when we incorporate other data, such as data from the transcriptome, gene expression, or data from the epigenome, the effects of the environment on our genes, and also perhaps data from neuroimaging. But we're a long way from a biological diagnosis of ADHD. Can ADHD genes, if you will, or DNA variants involved in ADHD predict response to ADHD medications? Well, I've only talked today about DNA variants that are associated with disease. There are also studies about DNA variants that affect drug response, especially response to the stimulants, methylphenidate and amphetamine, where there's the most work, but also some for drugs like atomoxetine, which is a non-stimulant, or extended-release clonidine, also another non-stimulant. And what those studies have shown is that there are fairly strong effects of some genetic variants on drug metabolism, and those might be theoretically useful. But in practice, most physicians are able to titrate their medications in a way that they can detect fast and slow metabolizers without much difficulty. Nevertheless, in some cases, some clinicians find that knowing about the genetic variants that control drug metabolism can be useful. Now, there are also studies that look at the effect of DNA variants on the pharmacodynamic, or if you will, the efficacy response of symptoms in ADHD. I recently published a review in Molecular Psychiatry about that, and we showed there that there were 5 DNA variants that were statistically associated with response to methylphenidate, the degree to which methylphenidate lowered ADHD symptoms, but the predicted value was low, so they really weren't very useful for telling doctors what's the best medication to choose. Ultimately, what we need are studies of what's called pharmacogenetically guided treatment to see if, when physicians have information about genetic polymorphisms, genetic variants, do they do a better job from clinicians who don't have information about those genetic variants. These genetic data with ADHD also tell us a bit about how we should think about psychiatric comorbidity. There have been, in the last decades, sometimes fierce debates about psychiatric comorbidity in ADHD. In the 1990s, many of my colleagues didn't believe that ADHD could co-occur with depression or bipolar disorder, for example. And then after there were many clinical studies and epidemiologic studies, those debates tended to reduce. It was only recently, until the publication of DSM-5, that clinicians were allowed to diagnose ADHD in the context of autism spectrum disorders, because because it was believed if you had an autism spectrum disorder, you couldn't diagnose ADHD. Now we know that's wrong. But these DNA data, these genetic data tell us that not only were those rules wrong from the perspective of the clinical presentation of patients, they were also wrong based on the biology of the disorders, because now we know that these disorders share some DNA variants to a highly significant degree. And so I like to say that diagnosticians should expect pervasive comorbidity, because ADHD can co-occur with many disorders. I've shown you some today, but ADHD also co-occurs, for example, with substance use disorders and antisocial disorders like conduct disorder and antisocial personality disorder. So multiple comorbidities are to be expected. It's the rule rather than the exception for the patient with ADHD. There are other diagnostic controversies that are very relevant to the genetic studies. The first is, now we know that common forms of ADHD, which means most ADHD, is a dimension in the population. And that argues against the idea that there are separate subtypes of the disorder that are somehow biologically distinct from each other. And in fact, there had been subtypes in the 4th edition of the Diagnostic Manual, and those have been appropriately retired in the 5th edition. There is some current talk about adult onset ADHD as being a distinct disorder, and that seems on its way to retirement, I hope, because the arguments about that were a little bit over-exaggerated, and we see from our own data that the genetics of adult ADHD are highly correlated with the genetics of child ADHD. We can also talk a bit about subthreshold ADHD. By that I mean the person with ADHD, the person who shows up in the clinic, or the child who shows up in the clinic and has a few symptoms of ADHD with some impairments that are associated with the condition, but they don't meet the full clinical diagnosis. These cases are very vexing to clinicians because they don't know what to do with them. What do you do if a child has 5 impairing inattentive symptoms and 5 impairing hyperactive impulsive symptoms, but they don't meet the criteria because they have to have at least 6 in one of those categories? Well, we showed, and this we is a group of us, me and some colleagues from the Massachusetts General Hospital in Boston, we looked at a really large sample, which was several hundred children with ADHD and children with ADHD, children with subthreshold ADHD, and children without ADHD. These were, if you will, typically developing children not selected for having psychiatric conditions. We look at a number of features. In the top left-hand corner, you can see the total number of other psychiatric disorders the groups have. And here you can see that the controls, of course, have very few disorders, but the subthreshold group in light blue has about the same number of disorders as the full threshold ADHD group in dark blue. And if you look to the right of that at the individual disorders, you can see most of the individual disorders, the 2 ADHD groups, subthreshold and full threshold, very similar to one another. Moving down to the bottom left-hand corner, we're looking at the clinical scales from the Child Behavior Checklist. These are ratings of psychopathology by parents, and also some social problems and other types of behavior problems. We see in all categories the subthreshold ADHD group looks very much like the full threshold group, and both are much worse than the control group. And we also see that in the far lower right corner when we look at the emotional dysregulation profile from the Child Behavior Checklist. So these are very fascinating data, very consistent with what we know about the dimensional nature of ADHD. These children that we call subthreshold are probably just below the threshold of diagnosis. It's like somebody having subthreshold hypertension or borderline hypertension. They technically don't have hypertension, but doctors worry about them because they're very close to the threshold. And such is the case with these children with subthreshold ADHD. There have also been controversies about the overdiagnosis of ADHD. Are too many children being diagnosed with ADHD in the United States? Well, this is a long discussion, which we don't have time for, but the point I want to make here is that one of the worries people had in the past is that, well, if ADHD is a categorical diagnosis, either you have it or you're not, a categorical model implies that a misdiagnosis is very serious because you're calling somebody and you're treating somebody for a disorder that absolutely doesn't have it. When in fact the dimensional model says many cases of overdiagnosis or misdiagnosis are probably people or kids or adults who are close to the threshold of diagnosis, and we put them on the wrong side of the threshold. And that's a less severe error, if you will, because it's very much like borderline hypertension versus full threshold hypertension. So I think in the future, I hope that studies of so-called overdiagnosis in ADHD will look at ADHD as a dimensional construct rather than a categorical diagnosis. The dimensional nature of ADHD's biological substrate, its genetic substrate, also suggests that this dimension might, as a phenotype, fluctuate, because as the child or adult with ADHD faces different stresses in the environment, they will show different levels of ADHD symptoms. So thinking of ADHD as a dimension will help clinicians think about how and why the disorder might fluctuate. For example, there are fluctuating demands on self-regulation. When a child is on the playground, there's less demands for self-regulation than when they're in the classroom. When the adult is sitting in a conference room attending to a presentation, there is more effort in the self-regulation of attention than when they're at home watching television. We also know that there's a developmental recovery of ADHD impairment and even some brain functions, and understanding how that change in ADHD symptoms is related to genetics and to the dimensionality of the disorder is a really important area for future research. So I think we'll summarize what I've said today about the genetics of ADHD in 7 main points. First, there's no doubt now from many, many twin studies, we have 38 twin studies, that ADHD has an extremely strong genetic component. We have now documented with genome-wide association study of a very large sample that genetic component has a strong polygenic etiology, where there are many, many DNA variants that increase the risk of the disorder. None of these variants individually has a very strong effect on the presentation of ADHD, but when they're combined in a cumulative fashion in conjunction with environmental risk, the disorder emerges. We've also shown highly significant, although small, genetic correlations of ADHD with other psychiatric disorders. This goes a long way to explain why we see so much comorbidity between ADHD and other disorders in the clinic, and may in the future help us understand the shared biology of these disorders. We also know that the genetic risk for ADHD is correlated with the genetic risk for other psychological traits and even medical outcomes like obesity. Another medical outcome I didn't mention was diabetes. The genetics of ADHD is modestly but significantly correlated with the genetic risk for diabetes. I've also shown you that the genetic risk for ADHD involves DNA variants that are highly expressed in the brain, and that many of these variants also are involved in regulatory regions, which tells us that we need to study how the environment affects these regulatory regions to change the risk for ADHD. When we look at studies of childhood and adulthood ADHD, we see that they too share polygenic risk, but that there's a percentage of that risk, about 40%, that's not shared. And that suggests that there are DNA variants that regulate the persistence and remission of the disorder. That's an area that's very important for future research. And finally, I've discussed some diagnostic practices, some ways that the genetic data can help diagnosticians think about the diagnosis of ADHD by using comorbidity, by expecting comorbidity in their patients, and by using the dimensional trait paradigm of ADHD to not only diagnose their patients, but to think about how their patients deal with ADHD as they face the fluctuations of stress and challenges to self-regulation in their daily life. And with that, I'll end, and thank you very much for your attention and interest in this presentation.